So welcome everyone. We are very happy to have you today for this last seminar of this special edition, March edition. But before that, we have to remind you that the Biomat Congress is coming very soon now, at the beginning of May, in Le Croisic, near Nantes. So if you plan to come, do not forget to register very soon because the deadline has been extended. But as you can see here, it's maximum March 31st, sorry. So please submit your abstract before. And also the whole program has been delivered, so you can check on the website the information for that. And so with this, I would like to introduce you the last talk of this particular edition, Women in Science. And so we have the pleasure to welcome Dr. Stéphanie Descroix, who is a research director in Institut Curie. And so she's going to present a summary of what she's been doing in this lab, so Cell Physics and Cancer Laboratory. And the talk is entitled Microfluidic Chips for Understanding Living Organisms. And so we have the pleasure to listen to you. The floor is yours. And
Okay, thank
I
you.
stop the share.
Do you see my screen?
Yes.
Okay. So, as Elliot said, I'm Stéphanie Descroix, and I guess you all recognize Narcisse. I will start with this a bit not very humble exercise to introduce myself and to speak first, I hope quite quickly about my career and my past. Okay, so let's start with the beginning. First, I had a bachelor in life sciences, so now I'm working in a physics lab, and I started mostly with life sciences, and then I moved slowly to biochemistry and analytical chemistry during my master. And finally, I did a PhD in analytical chemistry at Chimie Paris. So you could see that one of my difficulties is that I like many different stuff in my life. And I'm still very convinced that in science, you can be focused and unfocused at the same time. Okay, then I had the chance to get quite early after my PhD a permanent position at Université Paris-Orsay as maître de conférence, assistant professor. And that was for me the first time because during my PhD I did a bit of teaching, but you know, as it is during your PhD, it's not exactly the same. And then I learned how to combine research and teaching, and I must say that at this stage of my career, I did not have kids, and I have a lot of time to spend in the lab, and that was good to be able to combine this and to be efficient in both aspects of my career. But still, I also realized that even if I really like the students and the teaching part, I'm still I'm more research oriented person. So I decided to reapply to CNRS. And then, despite the fact that I already had got a maître de conf position, I had the chance to get a CNRS position as a chargé de recherche. And then I joined ESPCI in a lab of analytical chemistry. And my project there was really to be able to introduce microfluidics in this analytical chemistry lab, given that I was really not an expert at all in microfluidics, but with the host lab, we are really the will to introduce and to combine microfluidics and analytical chemistry. I had the chance during this year to meet amazing people, especially in the team. There were people from Pasteur Institute, so more life science people. And for me, it was super good to just connect again, to be connected again with more biological questions. I learned a lot, but I also learned that there are some environments in which you can evolve positively, and in some others, you do not fit with the environment you were arriving, and then at some point you have to be brave and to accept that it's not feasible, and then that you have to move. And I also, not only for this, but for many times, Institut Curie asked me if I would be interested to join. And I was really considering that, given the fact that life sciences is still something very important to me and my initial background, I would like to be closer to the biology and to the clinical application. That's why, after 7 years at ESPCI, I moved to Institut Curie. Bon, okay, it's a normal way of CNRS evolution in terms of CR2, CR1, whatever. And then some of you are very young, some not. So you all know that in your life and in your career, sometimes you meet people that help you to build the next step, and it was the case for me. Because Anne-Marie Gay, who was of the GDR Micro and Nanofluidic network, proposed me to take over after her, and it was great because it was really it took a lot of time, but at the same time, the time that you bring for the collectivity, and also a way to connect people, to meet new people, that was really a rewarding collective task to me. Then after I become research director, this we don't care. Another important point, at some point we founded a company, so I will go back to this very briefly later. That was also something completely new. At this stage of my career, it was pretty clear that it was not my main focus because I was about 40, and then I was really at this stage where I would like to enjoy as much as possible to be in the lab. So I just told them from the beginning that yes, this company is also super important, but it will never take more than 10% of my time, and mostly on science because I'm not a business person. And then a few years after, I took over also another person that was important to me, Jean-Louis Viovy. After the retirement of Jean-Louis, Curie is a bit special. Usually when the PIs get retired, the team is completely dissolved. It was not the case because they were considering that our activity should be maintained. But to become PI of this team, I had to go through a real competition with international jury and so on. And it was very good because I think for me, it's very healthy to not just consider that you are here, so you should be the next one. That good that you are ready to think on why it should be you, what you would like to do in your next step. And then it's a new adventure that is now a few years ago that I started and that I really like. Not super easy from time to time, but really cool. And then since a few years, and I think it's because you are getting older, people also propose you more collective responsibility. So I had a few new ones regarding IPGG and a PEPR. Most of them are very collective. And again, this is also for me at least, the beauty of our work, that I think in science, very quickly you understand that you can never do something just by yourself. You need the knowledge of other people. So being able to put people together to make them work, to see emerging new ideas because people are together, it's really something strong to me. So even if it's quite heavy, I'm really happy to have these other responsibilities and also new scientific opportunities. And again, it's collective. You could see it's not a one-person ERC, but with super cool and super smart scientists. And that's it. And then just to tell you that, of course, life is not always linear. And now I just show you the places where I were. Of course, you can interrupt me if you have questions, guys. Now that I have seen almost all your faces, do not hesitate. Okay. If yes, do not hesitate. Or in the chat if you prefer. Just to show you that the way, at least, I don't want to say that I'm an example. I don't think so. I think we are all examples just for ourselves. But in my case, my scientific life, my life as well, but it's not the question here, was not exactly linear. I really started in the late 90s, so I tried also to put some music to give you the song of my life. Let's see. I really started with analytical chemistry and something very specific on electrokinetic separation. And it was really a basic science driven. I had a wonderful PhD supervisor, really eager to work on understanding. So that was very good, but very basic, very fruitful. I had a very good, and then during my PhD, I had also the chance to get an Erasmus, so I spent a few months in Geneva, and that was very good. Then after, I move in terms of topics, but not so far. I still keep this analytical aspect, electrophoresis, but putting them at microscale, working with microfluidics. But then more and more, given that my initial background in life sciences, I started to shift to more biological questions. So moving from separation science, I moved then to microfluidics and bioanalysis meaning that you work with different questions of diagnosis, so biological molecule and even biological cells. We have been working with circulating tumor cells. And then now for more than 10 years, I completely stopped this activity of analytical chemistry or bioanalytical chemistry. And I went back to something that was maybe more precious to my heart, is more the interface between technology, biology, biophysics, and clinics. And if I put also Georges Brassens and Bob Marley, is it at some point I tried to check for the music at this period, and then I realized that at this period I was already old and just looking for my old singer and not to the recent one. That's why. Okay, so the core technology of my work is about microfluidics. I don't know if I need to introduce you what is microfluidics, but I will very briefly. So first, you could see a few examples of microfluidic devices that we built and design as a function of our questions, our needs. So microfluidics is both a scientific field and a technology that is interested in the manipulation of fluid, that could be gas or liquid, in tubes, channels in the range of in the micrometer range, let's say. So why is it interesting? It's interesting first because the physics at this scale is completely different, not completely, is significantly different from the one at the macro scale. And also because if you are interested in life science question, it's really a very good scale also to manipulate cells, single cells for sure, but not only single cells, but also aggregate or tissues. So I will just start to briefly show you what I did before, and then I will spend more time to explain you what I'm currently doing. So initially, my work on the bioanalytical aspect was a lot in the field of the combination of magnetic particles and bioanalysis. So magnetic particles, and I think I don't need to explain you what are magnetic particles, but maybe what I can tell you is why we were doing this and what I like in this work. It was already at the interface, and I think that my scientific positioning is clearly at the interface. Because here you need to understand the physics to build the system that you will need to use for the bioanalysis. So you need, of course, to understand all and this is also one specificity of microfluidics. Microfluidics, it's a matter of microfabrication, materials, biomaterials, surface chemistry, hydrodynamics, imaging, and then physics in terms of, in this case, in terms of magnetic particle organization. So you could see a few examples of what we have done, and we have, because it's really a team effort. At this stage, I was working a lot with Jean-Louis Viovy and Laurent Malaquin. I think we are really a trio. So we have been working on different stuff. So for instance, these images, I hope you see my mouse. You could see some bits that are here, self-assembled. So in these devices, for instance, what we did is what we just injected magnetic particles, and there were some stamps to allow the columns to grow. Then we apply a magnetic field, and the column will grow everywhere, but they will be stronger where we add this magnetic stamp. And then just by applying a flow, we remove the extra one and we just keep this one. And what is special is that at the surface of this magnetic particle, there is an antibody that is specific to the cancer cells. And then we were using this system, so it really started with physics, self-assembly of magnetic particles, to something that at the end has been used and validated with clinical sample. So the idea is that during the metastasis process, the cancer cell, they have to move from the primary tumor to the metastatic site, and they do so by blood vessels. And you can find these cancer cells in the blood of patient, and it is related, and it will give information to the prognosis of the patient, and then it could also help to select the proper treatment for the patient. But the problem is that they are at a very, very low concentration. It could be in the range of a few cells per milliliter, a few cells, given that you have million and billion of white and red blood cells, or I don't know, vice versa. So with that, we were able to flow inside the microfluidic devices this sample that was diluted blood. And then with that, we were able to capture and then analyze these circulating tumor cells. And you see really it's a mix between physics and bioanalysis. You could see also this beautiful system. I really like this. It's called a microfluidic fluidized bed. So here what you could see are magnetic particles. At the bottom here of the chip you can't see, but there is a magnet. And these particles, they stay packed, but you could see that there is a recirculation flow inside the microfluidic devices because they are subjected to two forces, the drag force in the direction of the flow in this direction and the magnetic force in the opposite direction. And if you tune your magnetic force and your drag force, there is a good balance between both of them so that you could get this beautiful system. And then again, these magnetic particles, you can functionalize them with any antibody, DNA, enzyme, whatever you want, to do bioanalysis. And for instance, we have showed that this can be used to detect, extract, and then detect circulating tumor DNA in this case. And finally, the last one, and I won't show much because I will just show a bit more after, it's something based on microfluidic droplets. Is it okay so far? Okay, I take the silence for yes. Okay. Then I will just give you a bit more details on the droplet-based assay very briefly, and then I will move to something that is maybe more of interest to you for the biomaterial question. I guess you don't know, but you are all experts in droplet microfluidics, given that you have already prepared a vinaigrette. So basically, this droplet microfluidics is just a matter of emulsion, except that you generate emulsion with a very well-controlled size and composition, and that your emulsion are quite stable. So in the field of droplet microfluidics, for instance, you can see here a way to generate droplet with content of two liquids, the black one and the white one. You could see that it's super easy to produce this droplet, that they have a perfectly monodispersity regarding the generation, but there are a lot of functions that people are able to do. You can split the drug, you can merge the drug, you can incubate the drug, you can here encapsulate single cell, for instance, inside this droplet. These droplets, they have a volume ranging from the picoliter to the microliter, and then they really, if you consider a microtiter plate, it's the same than a well, except that you can have millions of them in parallel. And as I mentioned, it's just very convenient for all the high throughput experiments you would like to run. But so far, most of these functions are super useful, but there is no extraction, no purification. It was really something missing in the toolbox of the droplet microfluidics. And now you could understand that we have for years a great interest in magnetic beads. And this is what we have done, and I will play the movie and then I will explain. What you could see is a droplet that contains magnetic particles, and in front of it there is a magnetic tweezers. And when you switch on the magnetic particles, you can quite easily extract the magnetic particles here. Okay, so then a bit of physics, and it will be the only one with a question. These particles that will be subjected to different forces. First, a magnetic force, okay, related to the magnetic field, the particle magnetization. But they will be also subjected to the drag force, because you could see that here the oil will push the droplet, and inside the droplet there will be some recirculation flow. And then the last one, if you would like to extract the particle, you have to fight again the capillary force, and especially the interfacial tension. So if the magnetic force is higher than the two other one, you could be in this situation where you extract a cluster of magnetic particles from a droplet that was initially containing the beads. And then after, it's super easy. You just have to remove them in another droplet that, for instance, contains your samples that you would like to use. I don't know. The first application that we had for that is to perform an ELISA, except that you perform an ELISA in a 100 nanoliter droplet. And in a few minutes, it was very short because all the diffusion distance are decreased. But with that, really, you can put the beads in different droplets as you would do by pipetting in your well. and then it was the first, and this is this paper, the old paper already. This was the first application of this magnetic tweezers. But then after, and very long story short, in one slide, we have been working in different type, both of application, physical and technological advances. So we have been using this to perform for clinical diagnosis of HER2 expression from breast cancer. And again, it has been validated in a blind study with patient samples. Something that we have been really successful with this droplet magnetic tweezer system is also to prepare DNA libraries for next generation sequencing. We have also shown that it can be used for protein digestion and extraction before coupling with mass spectrometry. And finally, we have been also working a lot with Leila Perrier and Céline Vallot on the use of these magnetic particles to perform multimodal single-cell analysis. But to do so, we have been working also with other type of condition. You see, at the beginning, we were just working with a tube and a nanoliter droplet, and then we moved to something very, very small with a few nanoliter and even less. I think we were in the range of picoliters, and integrating this in a micro on all dynamics of the system. And it was working so well that we were convinced that it can be useful for others than just us. And we had at this period very cool people and very smart people who are really ready to go for the startup world that is a completely different world from the one of academic science. And these guys, they founded like now 10 years ago, this company in Orevia that has had very high period, very low as well, and that is still around, and now that is selling a very beautiful machine. And just to tell you, as a scientist, it's super, super satisfying to see that your prototype that was efficient is able to become an equipment that others can use and for which they will really have an advantage. I mean, it's really something that gives you the feeling that part of what you do could have a short-term impact. This is not the only impact to which we are interested in, but this is one of them. And at the same time, it's also very rewarding to see that the people with whom you have started this adventure have been able to grow and become manager and CEO and CSO and blah blah. And then, of course, there are difficulties and obstacles, but I must say that I was really impressed by what they were able and by what they are still able to do. So if you are looking for a machine for preparation of library and stuff like that for NGS, just discuss with them. Okay. Okay, it was for yesterday, and now let's move to what we are doing today in my team. So in my team, we are interested in organ-on-chip technology. So I guess that if you are all from the biomaterials science, you all know that in biology we are using models. Initially, a lot of 2D models in plastic dish. Of course, we are also as a community using animals, and we also know that some of them and most of them, all of them, I would say, have advantages and limitations. So given this limitation, there was clearly a need for new type of models, maybe with an in-between complexity between the 2D and the animals. And among this model, and if you want, we can discuss and compare also, of course, you all know organoids, but me, I have been interested in organ-on-chip. That could be also, you will see at the end of the presentation, organoid-on-chip. So the idea of this organ-on-chip is really to recapitulate in these microfluidic devices the different feature of an organ that could be the 3D architecture, the different cell types, the physico-chemical environment, the function, or the forces that the cells are experiencing in vivo. Just I had a look to the time. So it goes with several advantages, and if you want, we can also discuss about the limitation. One of them is the miniaturization. For instance, we are working on a gut on a chip. In our case, I would say that the surface of the gut that we develop should be like 2 or 3 square millimeter. so way smaller than a normal uh, organ. And you will see also that in the presentation after we work with patient sample, and of course when you are dealing with precious samples, this question of miniaturization could be key. But for me, the beauty of this system is really the engineering aspect, and I think this is really something that we share with your community of biomaterials. Because as you are the one engineering the system, you have really the control over your cellular microenvironment. And if you would like to study the effect of the matrix composition, you change it. If you would like to study the effect of the spatial organization of the cells, you can change it. If you would like to study the effect of oxygen level inside the chip, you can change it. Meaning you can doesn't mean that it's easy to do, but still you can. Another point also compared to animal models is that it can be fully humanized. And of course, in animals, we can do intravital imaging, but then here with this transparent chip, of course, it's way easier to get the cell dynamics in 3D. So in my team, we are working with different type of organs, not all the organs of the human body, of course, because we have specific question about each of them. The first one, and that is for me the most difficult one we have been working with, was the gut. And you could see that for each of these studies, this is something that I shared mostly with biologists, but not only. And in this case, it was with my dear friend, Daniela Wisniewicz. We also had the chance with Sylvie Koskoy to work with Frank Bien-Aimé, and this is mostly Sylvie in this case, I must say that I'm more stepping back in this study, that Sylvie and Frank are working on a very specific pathology of the kidney, and we have developed with Sylvie this kidney on a chip in which you have tubes here that are hollow tubes covered with renal cells that mimics a nephron in a collagen matrix here populated with fibroblasts. Okay, we'll stop this turn. And then we have been also working with muscles more on the biophysical aspect, both with Edgar Gomes in Portugal, but also with Claire Willem in my team. In one case, we were working on the spatial organization by microfabrication, and with Claire, as she's also very interested in magnetic but nanoparticles in her case, working with magnetic bioprinting. So we had beautiful, not only images, we had a beautiful result for both types of approaches. And something that also is strong to me and for which I'm working for now years, more than 10 years, is my collaboration with, again, my dear friend Maria Carla Parini. With Maria Carla Parini, we use this chip to recapitulate the tumor microenvironment, and this is the first thing I will speak about in the presentation. And I will try to go fast because if we want to have time for questions. Okay, just to tell you, you certainly know that when people want to fight against cancer, they do not fight only against cancer cells. They have to take into account that a tumor is a very complex ecosystem with a matrix that is stiffer than in normal condition, less oxygen, different blood vessels, fibroblasts that are cancer-associated fibroblasts, immune cells. And to study these both in terms of basic science and application, we work with this reductionist approach of the tumor-on-chip development. Okay, I will go fast, but just to tell you that, and I think maybe that's the same for you in the biomaterial field, to develop something that is really part of our identity is to say we have biological, we have biophysical question, but we are really convinced that to address this question, we need to develop the tools and the approaches. So we are also very comfortable with the engineering part of our work. And this is what we have done with the positioning of gel, with positioning of cells or control of oxygen inside the chip. Okay, I won't tell much more than that. In these tumor-on-chip approaches, there are basically two main approaches. The first one is to say that is not exactly the same one than the organoids, but still in the same philosophy, to say, okay, I co-culture the cells in a 3D matrix, but I let them self-assemble. The second one is a bit different. It's to say, no, 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 no. The cells, they have a very specific organization. There is a scaffold with a 3D structure. I need first to reproduce and to provide the cells with the perfect geometry of the organ, and this is my approach. I will show you. So here, for instance, what you could see, and I'm sorry, I understood that you were interested in bone, and we did some stuff on bone metastasis, but I won't speak about it today. Okay, so the first approach that we have been working with Maria Carla for years is this question of I put the cells together in a 3D hydrogel, so 3D biomatrix, and let's see. No, not only let's see, it was a way to, this approach was used to decipher the mechanisms and then to move also toward clinical application. Okay, so this is the type of devices that we could bring to the community. So this is a chip here, a schematics, but this is something that we have been able to do, where you put inside the chip, here mostly, in our case, mostly we are working with collagen type 1, the cancer cells in green, the cancer-associated fibroblasts, the immune cells, and you can also put inside the system or built inside the system blood vessels, really with a cylindrical shape. And okay, it's not something that we have been reproducing millions of times, but once from the same patient, we have been able to extract and to get the cells, the cancer cells, the CAFs, the endothelial cells, and the T cells of the same patient that we have put together with this specific 3D architecture. And with Maria Carla, we have been working on different aspects. I just put the drug. Of course, it was just not a matter of testing, but better to understand to which extent we were able to mimic the mode of action of the drug inside this and to understand the effect of the TME composition of the mode of action of the drug. And I will just show you one example. This is something where we are working on a lung on a chip. And we had questions about immunotherapy because we know that with immunotherapy, the people who get a very good answer for them, it's amazing, but only 20 to 30% of the patients are good candidate and good responder. So there is a question on how the tumor microenvironment composition could affect the response to immunotherapy. And to do that, we have first been working with cell lines. Here it's an autologous pair of cancer and T cells from the same patient working with different ratio. And this is what you could see inside the chip. We put them in 3D inside the chip, and we had a dye so that when the cell die, they will just turn green. Super simple. And what we did is that we can first check for the viability of the cancer cells. Okay, when they are alone in the gel, it works pretty well. Then when you put the T cells, you could see a decrease. Oops, pardon. Uh, significant, but not 100% of decrease. And then if you use the immunotherapy, the nivolumab, the anti-PD-1 in this case, you could see even that there is, as expected, I would say, a response to the drug. But what is really beautiful that you really have access to all the ecosystem variation during the time course of the experiment. And with that, we have been able to show that this increase in cell death is also correlated with the change of the number of interaction between cancer cells and tumor cells, that you increase the interaction time, the number of interaction, and even you change the directionality of the T cells. Then we wonder if the composition of the tumor microenvironment would affect the response to this immunotherapy. And to do that, we have been adding inside the system this spindle-shaped cell that you could see that are cancer-associated fibroblasts. And what we showed is that in this condition, you completely lose the effect of the anti-PD-1 drug. So this type of data is a way to show that with this tumor-on-chip system, you can play with the different parameters to better understand, in this specific case, the resistance mechanisms of immunotherapy. Then I would like to just move a bit and show you that besides this question of basic science, and I just show you one example, we also have been working on the possibility of using this tumor on chip for clinical application. And it was done in a specific case, breast cancer. I will go fast on that because what you have just to remind in that, okay, breast cancer, you all know that usually breast cancer, you have a very good prognosis because like 80 or 90% of the patient are still there after 5 years. So it's a quite good I would not say good cancer, but you understand what I mean. But the problem is that when you reach the metastatic, if the patient reaches the metastatic stage, in this condition, the efficiency of the treatment is way lower with only 10 to 30% of response rate. And the other problem that the clinicians are facing is that at this stage, they have a few drugs that they could use for the patient, but there is no rationale to select the one that will be the most useful. No biomarker and no rationale. So we were considering that maybe there is something we could do for that, but at the same time, we had also to understand that, of course, you just think that your system will be perfect to address this clinical question, and then you realize that beside the question, there are also a lot of challenges. One of them is the type of tumor tissue that you could use. If you want to use this in the clinical practice, you have to work with biopsy. So first, it's not so easy to access, but it's also not easy sometimes for the patient because it could go also with pain and complication. And then the amount of cells in a biopsy is so small that it's another challenge. And then finally, something that is really also important, moving to this clinical application, is to consider, am I able to provide a result that will make sense for the clinician in a given time that will be also meaningful? So given that the patient could go for chemotherapy every 2 or 3 weeks, you should be able to provide the result in this time frame. So in our case, we were considering that we had to provide the result before 10 to 15 days, starting from the sample. So to do that, we have not been starting, and I think what I like in these studies, it's also an example to show you that sometimes you do not start with the most difficult part, but you go step by step. We have not been starting directly with the patient sample. This is now something that we are doing, but I would just like to start with the beginning. We have been starting with animals, so especially with patient-derived xenograft. That has been shown in the case of this breast cancer that it's a good avatar, meaning that if you treat the patient and the PDX from the same patient, most of the time they respond the same way. So our point of view was to say, if the PDX respond like the patient, let's take the tumor from the PDX, put them on chip, test the drug, and if we have the same response in the PDX on chip and in the PDX, we could expect to have the same response from the PDX, from the tumor on chip and the patient. Of course, it was a lot of optimization and process development. We have been working with different type of breast cancer subtype, different type of drugs, all in the standard of cares, but chemotherapies, antibody drug conjugated. It took us also a while to be sure that what we will provide as a response was strong. So we have been working with replicate, technical one, biological one. Okay. Okay, I can skip this. And basically what we have done, and I won't go through all the details, but we have really been extracting the cell viability of the cells inside the chip as a function of time for the different drugs. And we have compared this to what has been obtained in the animals in the PDX. And first, what is important to say is that with one PDX, we were able to do like between 30 and 50 chips. So first, it's higher throughput. Another important aspect is that maybe you could appreciate here is that in 4 days, you could see differences between the drug for one specific year sample. So it means that our time to results was about 4 days. That was really compatible with the clinical practice. And then we run this type of experiment with different type of drugs, as I mentioned, for different types of PDX. And then you could see with this heat map of the drug response as a function of the drug concentration, that basically you just have to appreciate that there are different colors, that meaning that at 4 days and in this condition, we were able to discriminate the sensitivity of the PDX. Okay, fine. But then the question is that this sensitivity, is it the same one that what we observe in the animals? And this is this table with this green and red colors that you can again appreciate, meaning that most of the time we had quite good correlation, and you can see here the number, and I won't go into details, but for the one that were red, it took us a while, but we really understood why, and I can detail, but I think we don't have time. We did the same with drugs that are a bit more complex, like this antibody drug conjugated. And it works also in the system, even if it was not perfect, given that we did not have, in this specific case, the immune cells from the patient. But then now the difficulty was moving to biopsies. And just to give you the flavor of what is a biopsy, here we have done that directly on a tumor. Here I think it was a PDX. And just to show you how small are the samples that you get. And then we had to completely revisit our system and especially to reconsider the chip that we were using. Initially we were using commercial chip with quite large dead volume, and we designed a new chip in order to make it possible to have from one biopsy, like depending on the number of cells in the biopsy, but 10 to 20 chip to test the drug. So I'm sorry, they are not the last samples, but now we have more than 10 patients, 4 biopsies. So it works. I don't know if we have a correlation with the patient outcome, and it will be the next step. So because of course it will take time, and this is something that we will launch in the coming months. I think it will be at the beginning of 2027. We will launch a clinical trial to really assess to which extent this tumor on chip can be a good avatar and help the clinician in their practice to select the best drug candidate for the best patient. And now I see the time and I see that I'm super late, maybe also because of our technical issues. And I would just show one images, just one, except this one. So I told you at the beginning that there are two approaches. The one is self-organization. The other one is based on the architecture, providing the cells with the architecture. And this is something that we did with Daniela and Luca. We designed a system like that mimic a column with script here. And we try to mimic colorectal cancer, meaning putting calf and organoids, so it's organoids on a chip, with mutation that will mimic colorectal cancer. And I will just show you what we have done with that because these images are so beautiful. With that, the question we were addressing is that maybe you know, but in your gut, there are muscles that push the food, and this is what we call the peristaltic motion. And at the level of the cells, it means that the cells are stretch, compress, stretch, compressed. And the question of Luca that we have with Daniela is that does the peristaltic motion affect the cancer cell invasion? And then we have designed this chip where you could see you have the cells here, the cancer cells in the crypt, surrounding by cancer-associated fibroblasts. And here you can appreciate the stack. And this is without stretch, and this is what you obtain after 24 hours of cyclic stretching, mimicking the peristaltic motion. So voilà, we are here. We are here in the sense that we are providing the cancer architecture with this crypt shape scaffold made again of collagen type 1 with a laminin coating to mimic the basement membrane. The collagen is populated with the cancer-associated fibroblast, and we are able to put the cancer cells at the surface of the gel to mimic what is supposed to be an epithelium, but here the cells are quite differentiated because they fill all the volume of the cells. And we showed that for this specific tumoroid with specific mutation, it seems that there is a strong effect of the mechanical forces regarding cell invasion. And that's it for me. And you could see, and that's super important, that is really an interdisciplinary effort and a very, very, very collective effort. And that it is really rich and fun to work with nice and smart people. And sorry, I was super long, so it's time for question now.
Thank you. Thank you, Stephanie. Yeah, so we have time for a few questions. People, if you have questions, do not hesitate either to switch your mic and ask it directly or write it in the chat. So regarding microfluidics or Stephanie's career, or anything you can.
Anything I could have a chance to answer.
Please don't be shy.
Can you hear me?
Yes.
No.
Yes.
Hi. First, thank you very much for your presentation. Um, I'm more interested in the microfluidic system for the cell culture. We try to do it in our lab to collect the extracellular vesicles,
Yes.
and we face some problem with the system that we have kind of some bubbles that present in the chips over the time when we try to culture them. Do you have any kind of advice that we can do to minimize the bubble air?
It depends. I don't know. I don't know your system at all. So it's not. Bubble is probably something that most of the people working with microfluidics faced at some point. But
Mm.
usually in static condition, it's not something you see bubbles already from the beginning or not?
No, it's when I start having the flow, when we
Ah,
put
yes.
like 10 microliter per minute as a speed, it's
I see
really
their speed.
low. Is high speed or slow speed?
Ah, it's high speed for me, 10 microliter a minute.
Okay.
So first, there are stuff that you could find that are bubble traps
Mm-hmm.
that
Yeah.
exist.
Yeah.
Then for us, the way when we had to put flow inside the system is the case for the kidney, and it's also the case for the blood vessels, the vascularized tumor on chip. The way
Mm-hmm.
we have less bubbles is to put the flow with hydrostatic pressure, meaning that we have a tube that we fill with liquid at a given height so that we can control exactly the pressure given that you know the resistance of your system. And with that, basically, this condition for which we have almost zero bubbles. And sometimes bubbles come also from the way you connect your system. It could also come from the surface property of your system. If it's fully hydrophobic, it's a way to nucleate bubbles. So there could be many reasons for that.
Mhm. Okay, because the system we have is we have the cell the medium culture in like a falcon tubes, and then we have one machine that have one connection to the tube to have the pressure, and it's gonna pump the media through the like a sensor and then a resistor into the chip.
And the chip is what, just a channel, or is it something more
It's
complex?
a channel for like a 2D system culture.
Okay, so normally, at first, I guess you try without cells
Okay.
to
Okay.
avoid to. And sometimes it's a matter of connection. It could be you could
Mm.
degas your system. I don't know which system you use to fill to inject the liquid.
Is the machine is Flow. I don't know if you know the
So I
system.
never use this
Mm.
one. I was using
Mm.
one from the competitor.
Okay.
But with this system, we have been working, I think, almost in the same condition. Now, this is something we are able to manage.
Mm.
So take your time, and sometimes it also depends on your material. Which material of chips are you using? But maybe we won't do a
Is
troubleshooting. But
IBD? That's the
yeah, okay, so it's plastic. So sometimes also
yeah.
coating could help to have something more hydrophilic.
Okay. Mm. Thank you very much. I'm taking notes.
Thank you. Do you have any more question in the chat? Last opportunity.
Yes,
Oh yeah. Go on.
if it's I can go. Okay, so I ask my question. First of all, thank you for your presentation. Regarding microfluidic, I would say I'm more in the beginner side of this science. So I would know if you have like general advices when one want to start to design such a device.
Yes, do not do everything by yourself because the difficulties that you will be facing, people have already faced them. So you will save time if you find nice people close to you who are used to work with microfluidics. Do not consider that all their solution will be yours because it's often that people will sell what they are used to do. Sometimes it's not fully adapted, but I mean, the problem is that, come on. Sorry, when you start with microfluidics, you will be facing difficulties sometimes with simple dust, clogging, bubbles, and sometimes you will have the feeling that it will never work and that you are wasting your time. Well, first, be brave. It's not the case, but sometimes it's just because you are not used and you don't have the very simple skills. So take some time, go 1 week in a lab where people are doing this every day, so that you will be really trained in the daily life of microfluidics. This is my main advice.
Thank you for your answer.